Okay, so in this video, I'm going to talk quite a bit about vision and try to give some lessons about sensation and perception in general, using this as a specific example. And then in the last video, I'll talk a little bit about different senses, focus a little bit on hearing, and get to a few others as well. So, vision, kind of for most people, kind of a ubiquitous sense. It really colors a lot of our experience, even colors the way that we talk about it with that. Uh, uh, Unintentional way that I used a visual sort of reference to talk about perception there by saying color. But there are some interesting things that you may have read about in your textbook already about how vision actually works. So when you see something, you're seeing light entering through your eye and through your pupil, which is kind of the aperture within your iris that lets light enter. And right behind that pupil is this lens, this little oblong, clear type of object that focuses that light and actually flips it upside down to project on the back of your eye. And the back of your eye is called the retina. And the retina is made up of these little photoreceptors that I'll talk about a bit more in a minute, called rods and cones. All these different little cells that can sense light in different types of ways. The center, the very back of your retina, has a particular concentration of these receptors. That's called the fovea, where most of the things that you see are going to, or the center of your vision rather, is going to project there on that fovea, so you can focus more directly on things that you want to. We also, in the back of your eye, the way this gets transduced into neural impulses is that these photoreceptors are sending signals that coalesce in the optic nerve, and that signal goes through the optic nerve, through your brain to your occipital lobe that is in the back of your head from each eye. So you can see here this pen image. When it goes through the lens, the light from that image actually gets flipped. So the light that gets projected on your retina, on the back of your eye, is actually upside down. So your brain actually has to do the extra work when those impulses get to your occipital lobe of flipping the image that you're actually seeing with your eye right side up again. Another really interesting thing is that right where the optic nerve enters your eye, there are no photoreceptors. So just below your fovea, where you have the most detail or ability to see detail, you have no ability to see anything. And it's a pretty incredible thing that when we look out into the world, our brain sort of just fills in the gaps. You don't see a dark spot of nothingness in your vision. Your brain fills it in for you, basically, because that would be kind of alarming to have a blind spot. But there are parts of your field of vision that you can't actually see right now, but you have the sort of illusion of seeing it to see this constancy of vision. I mentioned this briefly in the biopsych lectures, but it's interesting to note too that your vision is kind of flipped in how you perceive it from what you might expect in another way. That the right field of vision that gets projected on the left side of your eyes here is processed mostly, not entirely, but mostly by traveling through your optic nerves to the left hemisphere. So the stuff you're seeing on the right side of your field of vision is processed mostly in your left hemisphere by that blue path. The red path here is showing the left field of vision being projected mostly on the right side of your eyes and going to the right side of your brain. Again, there's no part of your brain that is totally doing one job, right? But there is localization in that there are types of emphasis that different parts of your brain are taking more of the load of, let's say. They're the leader in a process that's going on. So just kind of interesting to note again how that is flipped in that way. And I also like to point out to people how narrow our field of vision is in another way. That the entire visible light spectrum of these frequency of light waves that we see, light particles are moving in a wave like fashion, and the frequency at which they are vibrating or having those waves determines what color it is for us. So light with higher frequencies seems blue, light with lower frequencies seems red, and there's all sorts of colors in the middle. But that is just this tiny, tiny sliver of the light spectrum, really. This is, on a exponential scale here, showing that we don't see X-rays or gamma rays or UV light or infrared light, microwaves, radio waves, long radio waves. All of those things are essentially the same thing. They are photons moving through the air in wave formats, but we can only see a little bit of them. That's just what we have adapted to be able to see. Other creatures, for example, might be able to see a broader spectrum of colors. They might see some UV light and some infrared light. And there's evidence that some creatures can even sense things outside of that area, maybe even to X-rays or microwaves. So we only see a tiny little sliver of the different lights that are traveling through our atmosphere. Okay, so the different receptors in our eye that I've mentioned, each one of your eyes, each one of your retinas, the kind of back of your eye, has 126 million-ish of these different photoreceptors. And these receptors that are kind of specialized are called rods and cones. There are many more rods than there are cones, on the order of about 20 times more rods than cones. Rods are long and thin. Cones are short, thick, and kind of tapered to a point. That's why they are called cones. Rods look like cylinders, like rods. Cones look like cones. The periphery, the outside of your retina, so the edges have more rods, and the center, or the fovea, where you see more detail, has more cones. And this is partly because rods are about dim light and seeing black and white, basically, seeing contrast. So your peripheral vision, it's much more important for you to be able to see contrast and make out shapes more in general. Your cones are how you see color. They're adapted to be able to see these differences in wavelengths in a way that rods are not. So in bright light, you're discriminating between colors, and that's when cones are being used more for that purpose. There also are 3 subtypes of cones, so we can tell the difference between different colors. Sorry, I thought I had something different here. So we also have this kind of these different types of ways that we detect things, high and low level detection. And low level detection would be just discriminating between bright or dark light or different colors, or if something is right in front of you or not. Feature detection is this high level detection. This idea that we have particular neural patterns and particular cells specifically in our eyes that are responding to particular events. So you have feature detectors, certain photoreceptor cells that are very in tune to being able to notice vertical lines. And they fire very strongly when you see a vertical line, but when the line is tilted, they might fire less strongly. And when you see a horizontal line, those particular cells might not fire at all. So we have these very specific feature detectors. We have even higher level detection than that, to see things, more complex shapes like corners or edges of things, or things in motion. And we even have some types of feature detectors that fire when we see faces, essentially, responding to ones that look realistic most. But even if you show someone a face with kind of mixed up pieces, they might fire sort of weakly. Or if you show someone a cartoonish face, the same types of detectors might fire. So this is an adaptation again because it's very important for human beings to discriminate between faces, tell who is who, be able to see different facial expressions on people. So we have adapted as a species to be able to detect faces in a very efficient sort of manner. Sometimes these ways that we detect features like edges on things can also be a little bit misleading though. So in this picture, I would ask you to look at the top part up here and the bottom part here, and kind of think to yourself what colors these are. This one you might be saying looks gray, and this one you might be saying looks white. And this is kind of similar to that checkerboard illusion I showed you earlier, where these things look very different, but if I again superimpose bars over the part where it looks like it's coming together, you can actually see that the top and the bottom here are exactly the same color. So the way that you see this sort of darkening and lightening difference here because of the shadows kind of tricks your brain into seeing differences here. So what's actually happening here is the real distribution of luminance or brightness is going from in the middle to darker, way up to lighter all of a sudden. And when you see these sorts of big differences between light and dark, your brain kind of exaggerates the differences before and after them. So you perceive this difference as being bigger in this top area compared to this bottom area because of your feature detection abilities to see the differences in brightness and curvature here. So your feature detectors, in a way, are kind of tricking you into seeing these as very, very different colors when they're really not all that different. The way that your rods and cones work, to kind of circle back, or your cones rather work, to go back to that, the way that we discriminate different colors visually, there are some different ideas about this. You have three different types of cones, you remember, and one theory of them, we have red, blue, and green cones, basically. And just like primary colors, I know it's green instead of yellow, and primary color is yellow, not green. But just like mixing colors, an early idea about color vision was that you just mix together these different cones, basically, and they are showing you the different colors. But there's a competing idea that really it's not that you're just mixing together these three cones, it's that they either fire in one direction or another. So yellow is kind of outside of this experience. It just has to do with red, green, and blue, really. But there are links between red and green, blue and yellow, and black and white. So you have these different photoreceptors that respond negatively to one color and positively to another. So the red-green receptors, if they fire positively, you see red. If they fire negatively, you see green. If they respond positively, you see blue. Negatively, they see yellow. So your perception of these different colors isn't really quite as independent. It's that seeing red actually makes it a little more difficult in a way to see green. So I want to show you this particular example, and I'm going to have you just focus on that center square, kind of widen your vision, get a little closer if you need to, and for about 15 or 20 seconds, we're going to focus on that center square. And you'll notice that the upper, keep focusing on the square, don't really look at what I'm doing, use your peripheral vision. But that red square here is firing your red-green photoreceptors in a positive way. The green one is firing your red-green photoreceptors in a negative way. So when I place a white field over this, you should see green where there is red, and red where there is green, and probably yellow where there is blue, and blue where there is yellow. So, did that work for you? You can back up if you want to do it again. It's kind of cool. But the reason that's happening is because your photoreceptors are working really hard to show you red, and when they stop working, they go from positive to negative. It's hard for them to go right back to the middle. They sort of overcorrect and go negative. So after you saw red, you see green. Another example, focus on this X in the middle, do the same thing, kind of keep your eyes open, and you might be, noticing, of course, that this is a weird color image for the American flag. It has the colors incorrect, obviously, but if we look at it for a few more seconds, and then I put a white field over this, because of this opponent process way that it seems that cones function, you will see the correct colors of the flag for just a brief second. So if you want to do that again, rewind a little bit. It's kind of fun, honestly. But these two things are evidence that trichromatic theory, where you're just mixing together different firing of cones to see different colors, is not correct. It's actually this opponent process theory seems to have much more explanatory power for how we see and experience colors because of what I just showed you. The way that we not only see colors, but the way that we see groups of things, this kind of difference between sensation and perception again, is largely influenced by how things are arranged in a way too. So you perceive things as being grouped in different ways if they are organized according to these what we might call Gestalt levels of grouping or Gestalt laws of grouping. So Gestalt psychology, this general idea that in a way, like things are more than the sum of their parts, and that you might not be able to describe something completely without describing relationships between things, in a sense. So the way that we tend to group things is by these different laws. Proximity, you don't see 12 dots here, you see two groups of dots. Similarity, you don't see 12 dots here, you see some white ones and some blue ones. Closure, you don't see 24 curved lines here, you probably see 12 dots because your mind is kind of helping you close these and seeing each one of these pairs as a singular entity. Continuity, you perceive a circle here, even though there isn't one, right? In fact, it's probably closer to a hexagon, but you might be perceiving this as a circle of dots. Connectedness, again, makes two groups appear, and if you have some sort of region having things together, then that makes them seem like a group as well. So even though there's not that big of differences between a lot of these pictures, it can really change the way that you see things as being related or not. Another interesting thing about how we recognize objects, we recognize things by their different component pieces that some people would call geons, this kind of cutesy word for that's short for geometric icons. So if I show you these two things, since you, at this point, you have no frame of reference, you have no top-down processing, you're just seeing some pieces and trying to use your bottom-up processing skills to see what is in these pictures. So it might be hard at this point for you to tell what they are. Kind of think to yourself and figure out if you can tell anything in here. If I add some more geons in here, maybe it gets a little bit easier, that bottom-up processing is matching up with your top-down processing a little bit faster. If I show you all of it, you can see there's kind of a wine glass or chalice or something and a watering can. But when you only see certain parts of those shapes, then it's really hard to put it all together. But the more geons you get, basically, the closer you are to being able to figure out exactly what you're seeing here. A couple other things I want to mention, these sorts of visual cues that we have. We have different cues for depth perception, motion, and constancy. So depth perception, we have these different cues, monocular cues, meaning use them with one eye, that things go to a point in the distance. This sort of linear triangulation that goes off in the distance is a signal that something is further away than something that's out wider in the picture. So we have those types of cues like this one, fuzziness, things being smaller. Those are all cues you can use with just one eye. Binocular cues, on the other hand, are things like retinal disparity. Binocular meaning two eyes, and retinal disparity meaning the image that you're seeing of something is different on one eye versus the other. And your brain kind of averages them to make you see it a little easier. But if you cover one eye, for example, and look at something, and then switch it over, or just close one eye and flick between your two eyes, you'll notice that something is moving. The closer it is, you put your finger in front of your face, that's going to move a whole lot. But if you just look at it, your brain kind of averages where something is. So that's another cue for depth perception that happens to be a binocular cue to using both of your eyes. Motion perception gets really complicated. It's really hard for you to just track motion across your retina. There's an example of this might be this thing called the Phi illusion, that you sense motion here, even though there really isn't motion. You see this absence of a dot moving in a circle, or perhaps even an incomplete circle turning, even though it's actually just that things are appearing and disappearing. And this Phi illusion is really how we can consume a lot of media, right? Movies and TV shows, for the most part, are shown in 24 frames per second, and that is sufficient for our eyes to kind of compensate for that and smooth the image so we can see a constant sort of motion there. Higher frame rate movies might sometimes look more realistic, and sometimes the complaint for those is that they look kind of fakey because actually they're closer to what our real vision looks like. So that's why things that are more accurately portrayed on film might look kind of fake in a way. and really the human eye, most people can really only perceive about 45 images per second. So anything above and beyond that is just kind of not really doing anything for most people anyway. We also happen to have feature detectors, those high level detectors. We have those for motion as well. So there are certain photoreceptors you have that are firing off when you see something moving. Um, Perceptual constancy is another thing. We, perceive things as being constant, even if we, don't always have them being that way. So if somebody is walking away from you, you perceive their size as being constant, right? You don't perceive them as shrinking as they're walking away. You know that they are constant in that way. And when, things that, if I take a pen and I hold it this way and I turn it, you don't perceive the pen as suddenly being very small. You know that I've turned the pen, right? So our brain is very good at filling in those sorts of gaps and telling us that things are constant in a way that we might not always realize. Like a door being shut. You know that this door is the same when it's closed versus when you open it, even though it looks very, very different in those different angles. One last thing I want to show you to kind of show you how you can trick these things sometimes is this kind of funny looped video I have called an Ames room, where you see that the way this room is shaped and set up, when these people walk through it, it looks like one of them is growing and one of them is shrinking, but really it's that this part of the room over here is much further away than this part of the room. Now I'm going to put up a diagram. The diagram I have is actually flipped, so don't get too confused by that part. But I want to show you how this really works. That if someone were peering into this room, they would see the guy in green would be standing down here, and the woman in orange or red would be standing up here. So they look like they're the same because of the way the floor is slanted, but they're actually in very different spots here. So with that, I'm going to move on. We'll talk in the last video about hearing and a few other senses and kind of wrap up sensation and perception.